So now it's my great pleasure to introduce our next speaker, Dr. François Robert. François did his PhD at the University of Sherbrooke with Benoit Coulomb, doing foundational work on RNA Pol2 initiation complex. He then went on to do his postdoc with Rick Young at MIT, and he developed the genome-wide chromatin immunoprecipitation technique, Chipseq. From there, he went on to take a position at the IRCM Institute in Montreal in 2003, where he's been a full professor since 2016. And there, he's been a mainstay in the chromatin and transcription fields with fantastic work on a wide variety of topics, including RNA Pol2, CTD phosphorylation, the mediator complex, histone chaperones, and some histone modifications as well. Today, I'm very excited to hear the story he's going to tell us, which is about how transcription and the chromatin remodeler Chd1 join forces to convey fact to genes. So please join me in giving a very warm, if virtual, welcome to François Robert.
Ben, thanks for the very nice introduction, and thanks for the invitation to this great seminar series. So yeah, so as Ben mentioned, I will talk about fact today. I'll try to summarize our work, trying to characterize how fact gets recruited to genes. Okay, so as you all know, so nucleosome represents a roadblock for machines that need to translocate on DNA, and that includes, of course, RNA Pol2. So for RNA Pol2 to transcribe, right, it needs to read the DNA template, and for that, it obviously needs to unfold the nucleosomes. But for transcription not to leave a big mess on the chromatin, these nucleosomes need to fold back in the wake of transcription. So that delicate process is achieved with the help of many different proteins, including at least two very important histone chaperones, spt6 and FAT. Okay, so these chaperones need to reach active genes to do their job. So in the case of spt6, it's pretty well characterized how this is achieved. spt6 binds to Pol2 directly. It has a SH2 domain that allows it to do that. In the case of FACT, it's much less clear, and there's been a lot of different proposals in the literature to try to explain how FACT joins Pol2, but this is sort of an unresolved issue at this point. So at least it was when we started this work. So so just to say a few more words about FACT. So FACT depicted here at the bottom is made of two subunits in yeast. What I'm going to show is done in yeast, in Saccharomyces cerevisiae. Okay, so in yeast, it's made of spt16 and Pol3. And as you can show here, these two proteins are essentially made of histone binding domains. So all it does is bind histones in several ways. Even the dimerization domain here involving in connecting the two subunits, we now know it actually binds histones. So it doesn't have any domain that would easily explain how it gets recruited to Pol2 or to an elongation complex, you know, which is not the case for spt6, as I mentioned, because of this SH2 domain. So to first start to tackle this question, we decided to do ChipExo of FACT to get a high resolution map of where it binds in the genome. So as you can see from these heat maps here, so both subunits correlate very well with Pol2. So these are all genes sorted by decreasing transcription rate or decreasing Pol2 occupancy, actually. So FACT occupies highly transcribed genes. This has been shown by others using other methods. So this is just confirming what we knew already. You see that the correlation between Pol2 and FACT is quite good. And if you look closely, you're doing metagenes, you can see that if you look at the details, the two proteins, I mean, Pol2 and FACT don't correlate if you look at this level of detail very well in terms of their distribution along the genes. So Pol2 seems to stumble on the +1 nucleosome, but once it passes the +1, it looks like it drives through the gene quite relatively evenly, whereas FACT binding actually peaks at all the dyads, right? And that fits very well with the recent structural work from the Karen Luger's lab, who showed that FACT actually sits on the dyad of a nucleosome. So this is where it cross-links the most, right? The ChipExo shows you the cross-linking points, essentially. So at this level of detail, while POL2 and FAC go to the same genes, the transcribed genes, their distribution along the genes is different, right? Okay, so FAC correlates with POL2, but it also depends on transcription for occupancy, because if you shut down transcription, and this is done here using RPB1-1, which is a POL2 allele that, when switched to high temperature, shuts down transcription. When you do that, you can see that POL2 occupancy on genes collapses dramatically, and so does FAC, right? So taken together, these two first slides, which recapitulates essentially what we knew before, that FAC occupies genes and it correlates with transcription, but it also depends on transcription. So then the real question is, how is FAC recruited to these sites of active transcription? So in the literature, there are many models that have been proposed to explain how FAC is recruited to gene, and they can be categorized into two broad categories. On one side, you have proposals that FAC actually binds POL2 or bind an elongation factor, so it's essentially a recruitment to the elongation machinery, right? So it's been proposed to bind the CTD of POL2. It's been proposed to bind the PAF complex. It's been proposed to bind some other elongation factor, even chromatin remodelers. And some people have proposed that this might be how FAC gets recruited to highly transcribed genes. On the other hand, you have papers that have proposed that actually FAC recognizes directly the transcribed template. And one way to do that is by recognizing a histone tail modification that is generated co-transcriptionally. There are quite a few papers that will be consistent with the scenario. And but there's also a huge amount of work, of in vitro work, that very clearly shows that for FAC to bind a nucleosome in vitro, one needs to somehow disrupt the nucleosome, actually partially disrupt the nucleosome. So you either need to get rid of part of the DNA, or introduce a nick in the DNA, add a protein that would distort DNA structure. You need to tweak the wrapping of the DNA around nucleosome for FAC to bind this nucleosome in vitro. So one possibility is that transcription, by distorting nucleosomes, will help to enhance the affinity for FAC and recruit FAC. So there's a lot of in vitro data suggesting that, and quite recently, my host here, Ben Martin, published a very nice paper providing in vivo evidence for that model. So what we wanted to do here is systematically go through these models and test them in a unified system in the same organism, because obviously all these papers come from different labs, different organisms, and so on. So we wanted to test them systematically in a unified model. So we tested a bunch of mutants essentially for POL2 and FAC occupancy by chip. Okay, so I'm going to go back to the second part later. I'm just going to summarize what we found here. So using a bunch of mutants, we essentially ruled out binding to the CTD as a major factor for recruiting FAC, and I'm going to spare you the data. Same thing for the elongation factors we tested, and that includes PAF. So in our hands, deleting PAF doesn't prevent FAC from being recruited to genes. But in the process of doing that, we also screened for a mutant of Chd1, the chromatin remodeler Chd1. And there we found a very interesting phenotype. So in Chd1 mutant cells, what you see is that FAC, it can still be recruited fine. So it still goes to highly transcribed genes, but its distribution along the gene is skewed toward the 5' end. Okay, so normally FAC, again, peaks at all the dyads quite evenly, but in Chd1 delta cells, and we also found the same thing using a point mutation in the ATPase domain of Chd1. So this phenotype really seems to come from the catalytic activity of Chd1. So if you prevent Chd1 from working, from remodeling chromatin, you get accumulation of FAC in the 5' end of genes, in the first few nucleosomes. And importantly, that happens without any major effect on POL2 transcription. Okay, so in these mutants, you actually uncouple FAC from POL2 occupancy. So we think this is the first very direct evidence for FAC interacting with chromatin as opposed to pol 2. Because if it was really binding pol 2 or the elongation complex, its occupancy in the mutant should track pol 2, not nucleosomes. But again, as I mentioned earlier, that's an important point to stress. So in these mutants, FAC and pol 2 still correlate very well. So FAC still goes to the same highly transcribed genes. It's just that its distribution along the gene is skewed toward the 5' end. So putting all that together, so we started to think that, you know, FAC gets recruited in a transcription-dependent manner, although not by binding to pol 2 itself. But then after its initial recruitment, its distribution along the gene requires the chromatin complex, chromatin factor Chd1. So we think of it as a two-step process: recruitment and then distribution. But you know, if it binds chromatin and not pol 2, so what makes chromatin recognized by FAC, right? Actively transcribed chromatin recognized by FAC. So as I mentioned earlier, one can imagine that histone modifications, you know, achieved co-transcriptionally, like, you know, histone acetylation or trimethylation of K36 or K4, you know, may recruit FAC. So we've tested these again systematically with a bunch of mutants, and we really cannot find any evidence that genome-wide there's a major, you know, contribution of these histone modifications on FAC occupancy. So the model that remains is the fact that transcription by distorting nucleosome may allow FAC to recognize its substrates. And again, this is supported by a lot of in vitro data and also by a paper from Ben Martin recently. So to look at that more closely, we decided to go away from ChipExo and start to use MNA's Chipseq. And the idea here is that, you know, by digesting chromatin with MNAs, we can generate nucleosomes, right? And then if we enrich by chip, these nucleosomes are bound by FAC, and then scrutinize the DNA that comes with it. We may be able to get a better idea of this actual structure, the structure of these nucleosomes that are recognized by FAC, as opposed to the bulk of the nucleosomes in the cell. So the idea is to look closely at the DNA fragments that come down and see if we can get an insight from those about what kind of disorganized nucleosome may be recognized by FAC. And because we have a phenotype with Chd1, we obviously also did these experiments in Chd1 mutant cells. So before I show the data, I want to stress one thing here, which I think is probably obvious for most of you, but I think it's still important to point out. When you do an IP, what matters, you know, what reflects the binding of your factor of interest is what is enriched in the IP. And what is enriched in the IP is not necessarily what's the most abundant in your precipitate, right? So if you get something super abundant in the input, it may still be the dominating factor in your IP. Although depleted, it may still be the most abundant, you know, entity. So in a Chipseq experiment, actually, most of your reads actually are background reads, right? And you normally don't notice that because those background reads are just scattered along the genome and they don't form any peak or anything. So you don't really notice it, but they still make most of the sequencing that you get out of your Chipseq. When you do MNA's Chipseq, it's different because your input material does have information. It does have a structure, right? Your input is not just random. Your input has a very typical shape of nucleosomes, right? So whatever you IP from an MNA's extract will contain these patterns of MNAs, right? Unless you are able to enrich to an extent that you completely deplete your immune precipitate from those very abundant nucleosomes in the extract. But it's never the case in reality. So I just want to stress that in these data I'm going to show you, one needs to focus on what's enriched and not necessarily what's the most prominent in the IP. And here's a little bit of data that sort of illustrate what I want to say. So here I'm just showing the distribution of the sizes of the fragments from these MNAs experiments. So in gray, you see the input, right? So most of the fragments from the input are nucleosomal size, as you would expect. This is an MNase treated extract, and we get a little bit of dinucleosomes, and that's just because it's the way we set up the MNase digestion, so we don't over digest, so we make sure we still have some dinucleosomes. Now the blue traces show you the IP of fact from these inputs in wild type or in Chd1 mutant cells. So what you can see is that small fragments that are subnucleosomal size are enriched, right? And also fragments of internucleosomal size are enriched. At the same time, the nucleosomal size fragments are actually depleted, right? To the benefit of those other size fragments. But at the end, they still represent the majority of the DNA in a sample. But I would argue that, you know, if you care about fact binding, especially fact binding in the context of transcription, what you need to focus on is what's enriched, what's not the most abundant, right? And then our argument for that is shown on the right here. So if you do the same analysis, but you focus on the reads that align to the transcribed gene, the most highly transcribed gene, you see that this is even more pronounced, right? So these small and long fragments are even more enriched relatively to the nucleosomes, which are even more depleted, right? So I just want to stress that, you know, one needs to focus on the enriched part of the features of the data, not the most abundant features, right? And this is shown perhaps even more systematically on this representation here. So I want you to focus just on this image here. It's just showing the SPT6 IP from these experiments. So here, all the genes are sorted from high to low transcribed. And the color blue here represents SPT16 occupancy, the average occupancy on the genes. And this average occupancy has been computed using fragments of different sizes, from small fragments to very large fragments. So if you focus on mononucleosomal size DNA fragments from these IPs, you see that they're scattered all over the place. They don't really show up because they're just randomly distributed all along the transcription space, right? So if you assume that fact, well, if you trust that fact binds to highly transcribed genes, then this really shows you that these mononucleosome-sized DNA fragments are not the ones you should care about, because they don't enrich where you expect fact to be enriched, right? So again, they still represent the majority of the DNA in the sample, but this is background and it's randomly distributed. On the other hand, the small fragments, and the intermediate size fragments do concentrate on the highly transcribed genes, as you would expect for fact binding, right? So now that hopefully I've convinced you that you should focus on those size fragments and ignore the mononucleosome size fragments, I'm going to show you the data. Okay, so these are 2D maps of these MNase seq experiments. So on the left-hand side, you get the typical nucleosomal pattern that you get with the input, right? Just to walk you through this in case some of you are not familiar with these representations, but everything is aligned on the plot diad of the +1 nucleosome. On the axis here, you have the fragment sizes, and then the intensity is shown by the color, right? So again, most of the DNA in the input especially is of mononucleosomal size, and the center of these fragments line up with the -1, +1, +2, and so on nucleosomes. And then you get a little bit of dinucleosomal size DNA, and this lines up in between nucleosomes, as you would expect. So now if you look at what happens when you enrich for the nucleosomes, that the particles that are bound by SPT16 or FACT, again, dismiss this because this is background. What you should focus on is the other small fragments, from which you don't learn much from these representations. I'll go back to small fragments in other kinds of analysis later, but in these analyses, what turned out to be useful is to look at the intermediate size fragments. So in that range, you get fragments that are enriched by FACT, right? And again, these map to highly transcribed genes, as you would expect, if you remember from the previous slides. So if you look at their distribution along the gene, you see that they create sort of an inverted V pattern linking the mononucleosome position to the dinucleosome, to the mononucleosome, and so on, right? And if you focus the analysis on the most highly transcribed gene, again, the mononucleosomes are even more depleted. These are even more enriched. So you see even more, obviously, these inverted V patterns. Now, quite interestingly, in Chd1 mutant cells, we get a very drastic difference, two differences, right? First of all, the signal is concentrated at the 5' end of genes and relatively depleted at the 3' end. And again, that fits the chip exo pattern I showed you earlier. So in Chd1 mutant cells, FAC is sort of stranded at the 5' end of genes. The other difference, which is quite cool, is that you lose half of those V's, right? So you see very well the stripe going up, but the downward diagonals are lost. And if you focus on the highly transcribed genes, you see these features even more apparent. You see clearly the stripes going up, but you don't get the stripes going down. And again, the signal is concentrated at the very beginning of the gene. So what does that mean? What do these fragments represent? So this is kind of abstract. So what I did is here, I drew a little diagram, try to schematize what those fragments really are. So here you see the V pattern, right, in blue. And here in red, I've drawn representative fragments along that diagonal, right? So based on their size and their location, this is what the fragments will look like. So you see that as you go up the diagonal, what happens is that the 5' end of those fragments is constant, but the fragments are increasingly longer from the 3' end, right? And now if you go down the slope, down the diagonal, it's the opposite, right? So the 3' end of those fragments is the same, but they get progressively smaller from the 5' end, okay? So now it's very tempting to imagine that these represent intermediate of a process of FAC, traveling on the gene. And if you're ready to accept that hypothesis for a moment, if you and you put those fragments in order on the scale of time, you come up with a model like that where FAC will inchworm its way down the gene, right? It will bind the first nucleosome and then extend at one end, and then when it reaches the second nucleosome, it will retract the rear end and to end up finding the second nucleosome and which inchworm its way down the gene like that. In the Chd1, if you remember what we observed, we lose the fragments of the downward diagonals, and we get less and less signal as we go down the gene. So it's very tempting from this to speculate that what Chd1 does, it helps in that process somehow, and because it's not able to be efficient, well, you get progressively less FAC making it to the next nucleosome, and therefore less and less FAC as you move from the 5' end to the 3' end. So our data is certainly consistent with this, and it would be very nice if we could experimentally test the time dependence of this. But this is practically not doable, especially in yeasts, because the genes are very small. So in a minute, the genes are being transcribed for most of them. And we don't have a way to synchronize transcription in yeast like you can do in other systems using things like DRB or things like this, right? We cannot really do that in yeast. And even if we could, genes are so small that to track process going down gene is very, very difficult in yeast. So we don't really have this luxury. So what we decided to do is turn to modeling. And so what we did, we initiated a very, very fruitful collaboration with Jane Mailer's lab. So Andrew Angel in Jane's lab built a mathematical model of this inchworm idea, right? So the model goes like this. So a FAC would bind the +1 nucleosome. It would extend in the direction of transcription until it reaches and encompasses the first two nucleosomes. Then it would switch to a retraction mode, retract the rear end, and the cycle will repeat like this. So he built a stochastic model defined by probabilities per unit of time, where all these variables can be played with, right? So binding, extending, switching from extending to retraction, and so on, right? So This is a movie that shows simulation of these models on the left, and on the right you see the you know what the data would look like if you were to virtually do an M&A-seq experiment of this. So you see, several simulations of FAC inchworming its way down the gene, right? And this is a long movie, so I'm going to fast forward for you. So you can see that when at some point you reach a steady state where the image on the right looks very similar to the actual data, okay? So these are actually final images, not high resolution of what we get. So that model recapitulates this, which we think recapitulates all the key features of the data we see experimentally. While other models, obviously we tested alternative models, never do as good of a job, right? For instance, if you allow FAC to bind internally as opposed to bind specifically to the +1 and then invade the gene, you end up with simulations that predicts that you should have less FAC at the 5' than you do at the 3', which is not something we observe. So essentially all the models, alternative models we tested performed less well than that one. So we believe that at least our data is consistent with that model, even though I guess it doesn't prove that it has to be exactly necessarily that one, but it's the most consistent model we can think of. But now that we have a model with different parameters that we can play with, we can use that model to predict exactly what Chd1 might do, right? So essentially what Andrew did is to play with all these variables, increasing or decreasing the variables for all the variables in the model, try to find what parameter he could play with that will actually generate simulations that fit the Chd1 data. And among all the tested simulations he did, impairing the extension rate was able to recapitulate most of what we observed in the Chd1 mutant data. So this is a simulation for reducing significantly the extension rate. So what you observe is that when you do that, in the simulations, you lose the downward diagonals, just like we see in the data. And you also get accumulation at the 5' end relative to the 3' end, which is the other feature of the data. So you might notice here a little glimpse of what looks like even longer fragments. This is something we were curious about. So we actually plotted the simulations to the full scale. And this is what you see. You get these higher fragment size that are predicted. And these in the model are due to the fact that because translocation of FAC is crippled, right? You get sort of a traffic jam, and you end up with fragments, large M&As protected fragments, because you have a traffic jam that keeps these long protections. So if the model is right, then we should observe these in the actual data, and we actually do. So we used to always look at our data up to 400, but if we plot our data to up to 800, we can indeed see that these, which we had not noticed before, are actually present in the data. So because those were not used to build the model, but actually appear in both the simulation and in the data, they actually represent an internal validation of our model. So we're quite confident that this model makes sense. Okay, so that explains how FAC, after it's recruited at the 5' end of the gene, may translocate down the gene with the help of Chd1. But how is FAC initially recruited to the +1 nucleosome? The initial question I asked, how it's recruited to highly transcribed gene, still remains, right? So how does it find the +1 nucleosome of the transcribed gene specifically to start with? So an answer to that, or at least a glimpse at an idea of that, came from investigating now the small fragments. Remember the chip assay, the chip samples from these M&As extracts did enrich the internucleosomal size fragments, and I showed you the 2D plots analyzing them, but it also enriched small fragments that are smaller than the size of nucleosomes, right? So we analyzed that in more detail, and for that we were inspired by this very nice paper from the Enikoff lab, who showed that from these M&As seq experiments, if you focus on those small fragments, you can actually detect and measure nucleosomes that are partially unwrapped, right? And you can even highlight populations of nucleosomes that are asymmetrically unwrapped either from one end or the other end. And the way you do that is that you use those particular size fragments. You need to focus on very specific sizes. And what you do is you look at this distribution of the center of those fragments. So if a nucleosome is asymmetrically unwrapped, the center of the MNase generated fragment will be offset relative to where they were if the nucleosome was fully wrapped. So you get a shift of distribution of the center of those fragments relative to the canonical dyad position where you expect those, right? So this is very useful and it works very well. So we did that. So let's look at the input first. So if you just look at just MNase, just the input from MNase, so the data is here is in red, okay, and can be fitted very well to a double Gaussian. So the blue is the fit, and the gray are sort of a deconvolution of the two Gaussian that makes this the double Gaussians, which represents the data. So the easiest is just to focus on the gray because it sort of deconvolutes the two populations of nucleosomes that are partially unwrapped from one side or from the other side. So they're present in the input. So the input contains +1 nucleosomes that are asymmetrically unwrapped from both sides. That's essentially what this is telling you. Now, if you focus on what happens when you enrich FACT by chip, you see that you highly enrich one of those two populations, and that's the population where the nucleosome is unwrapped from the TSS proximal side. And we know that when Pol2 approaches a nucleosome, it does unwrap nucleosome from this side. So what we think is going on is that, it's transcription, again, that brings in FACT. And the way it does it is by, starting to peel off the DNA from the +1 nucleosome. So that gives an opportunity for FACT, to find a nucleosome, sit on the dyad, and then with the help of Chd1, it will make its way to the next nucleosome and so on, probably, in phase with Pol2, and, so that every nucleosome can be dealt with by FACT with the help of Chd1 as transcription occurs. So this is our model at this point. And so this is the summary. I can very quickly go through it. So through a combination of experimental and math modeling evidence, we propose that FACT is recruited initially to the +1 nucleosome, and then it needs Chd1 to move down the gene together with Pol2. Okay. So this clarifies how FACT interact with genes, but more importantly, it provides a framework to actually try to dissect the mechanism of all this, right? So how histone chaperoning occurs during transcription, and now we can introduce Chd1 in the picture and try to dissect in more detail what's the role of Chd1 in that process. So I think for all the biochemists, chromatin biochemists out there and structural biologists, these I hope will provide ideas for them to how to approach these things. So I would like to finish just by saying that this model may actually explain how FACT is recruited to chromatin in other contexts, because I've been focusing on Pol2 transcription, but FACT is involved in Pol1 transcription, Pol3 transcription. It's at the replication fork. It's involved in DNA repair. So it's not a specific factor for Pol2, just like a CT6 might be. So it makes sense that what it recognizes is not any particular feature of those machineries, but something that would be common to all of them, which, nucleosome unwrapping is obviously something that happens through all these processes. So this idea is very in line with in a hypothesis that was put forward in a very nice review from Tim Formosa and Fred Winston just recently published in NER. So what they propose is that FACT is actually a chromatin repair factor. So it sees, disrupted nucleosomes, binds to it to fix it, right? So in the context of transcription, we think this is what's happening in the +1 nucleosome, right? Pol2, as it engages the nucleosome, sort of disrupts it a little bit, and FACT joins. The twist to this from our work is that once that happens at the +1 nucleosome, this is not the end of the story, because you need more than that. You need more than just transcription to distort nucleosomes to bring FACT, right? that works for a +1 nucleosome, but for FAC to go to the subsequent nucleosomes, you also need Chd1, right? So it's not like as if any nucleosome along the gene would be recognized independently by a new FAC protein and so on. We think this is actually more coordinated with transcription than that. FAC would be, according to our model, FAC would be recruited to the +1 nucleosome through one of the, through this idea of having a disrupted nucleosome. But then it needs other activities to spread down the gene. And for these other processes, it might be other activities that help FAC to spread, or maybe in some cases it doesn't even need to spread. Like for Pol3 transcription, the genes are so small that there might be, just the initial recruitment might be enough to do the job. All right, so I just want to finish by acknowledging the people who did that work. So most of the work, almost the entire work, was done by Celia Geronimo in my lab. And the sequencing data analysis was done by Christian Poitras, a computer analyst in the lab. And I want to highlight again the fantastic contribution of Andrew and Jane, without whom, we would not be that far in our understanding of this whole process. And I want to finish by thanking you, and I'm open to questions. And as you can see here, we're happy to, host postdocs who want to join in our lab in Montreal.
Awesome. Thank you so much, Francois. That was just beautiful work. Great presentation. So we've got a number of questions. So I've got a question to start off here from Gregory Bowman, who asks, great talk. Does Chd1 help to evict FAC from the +1 nucleosome to help FAC move to the next unwrapped nucleosome?
I wish I knew what Chd1 does exactly. And I think, it's people like like you guys who can do that. What I can tell is that from the profile of the MNA's protection, and from the prediction from the model, it's really the extension part of the protection that is stimulated by Chd1. So I don't think this is pointing to Chd1 helping to release FAC, but rather to allow FAC to, and the FAC nucleosome complex to be remodeled in a way that it will eventually contact the +2 and the +1 nucleosome simultaneously. So it's that initial part that seems to require Chd1, not the second part, which is letting go of the +1 to concentrate on the +2 and so on. So according to the model, it would be rather the initial part, as opposed to letting go the initial nucleosome that would require Chd1. But one would need biochemistry to test that for sure.
All right, and then Carolyn Luger has her hand up, so I'm just going to let you unmute yourself now. You can ask your question.
Yeah.
My microphone is really bad, so can you read it? I put it in the chat.
Sorry, Carolyn. There's a lot of distortion. I didn't hear.
I put it in the chat. Can you read it? My microphone is super bad.
Yes. Let me read it. So she asks. Well, she had two questions. She wanted to first know whether you had read all of the books. Just behind you, and then on a serious note, she says, "How is the inchworm model reconciled with the structural information?"
Well, the books are. I haven't read them all, but I need to dust them every day, and that takes a long time. Actually, I think the model fits very well with the structure. Many features we see fit well. First of all, the fact that the main cross-linking points are at the diet fit very well with the model. Obviously, the model, not the model, but the 3D model that Caroline made, has to represent one. It's a snapshot of one step of the process, right? So which one it is? It's probably one of the initial part where FAC originally find one nucleosome, and then this will need to change as Pol2 progresses in the nucleosome, and so there's no inconsistency there. I think it's all consistent now. Which part of what we observe is actually pictured in the structure, not clear. Probably the very first step, I would say, when FAC first sit on the diet, it would be very interesting to get structural information on a structure that involves FACs binding 2 consecutive nucleosomes, which we predict is one of the steps of the process. So I hope this will come from you, Caroline, or someone else. But yeah, I think this is consistent. I'm not sure what else to say than to say that I think it's consistent.
So, okay. So there's a ton of questions, and so I'm just going to ask one more, and then we have a coffee chat afterwards. So for everyone who wants to join, we can chat more about the research and about how everyone's doing with the pandemic and everything else. So let's ask this question from Ivano Mochavini. Says that I noticed that the Chd1 mutant input nucleosomes are skewed towards the conformation with respect to the one that SBT16 prefers. Does this explain the phenotype of SBT16 binding upon Chd1 depletion?
Okay, so if I understand well, so he's saying that the profile we see in the input is also in the IP is also visible in the input, which is the case indeed. But it's highly enriched in the IP versus the input. So if it's in the IP, then it has to be in the input to some extent, obviously. But you're right; it is present to an extent that we actually can see it. So this is probably very, very prominent, especially when you focus on the highly transcribed genes. So I don't know what it's telling us, other than the fact that this is a very prominent, a prominent fraction of the chromatin that is stuck in that conformation in these cells, especially at the highly transcribed genes, which makes sense because this is where there's a lot of FAC business going on. I'm not sure that answers the question, but we can discuss that later further, I guess.
Okay, fantastic. So, so with that, I just want to thank both our speakers again for 2 fantastic seminars.